All right, let's hit the basics of fungi in this particular pre-lecture. So, when it comes to fungi, it's actually the greatest biomass in the world. They make up a huge amount of biomass on our planet. We have as many as 5.1 million different species that we've identified. More get identified all the time. Now, these are not necessarily singular cells, they are multicellular, but we tend to think of them kind of in the micro range because they are a little bit smaller. They're pretty ubiquitous. They're primarily found in soil environments. Just like bacteria, there are relevant fungi that have positive and negative effects on the human health. And the big difference that we're going to see from bacteria, obviously, is that they're going to have cell walls, they're going to have a defined nucleus and defined organelles. So you can see the picture kind of behind my head is a lot of different types of fungi. They have very various structures, which is really interesting. That makes them very interesting to study. So the primary role of fungi tend to be as decomposers. They basically break down dead matter, which is great. They are really, really essential on Earth because of that. And they're actually very useful in industrial processes, many times as fermentation. So if you think about fermentation processes to yeast, right, to make bread, is one great example. And they actually have been used to create lots of antibiotics. So penicillin, as the first antibiotic, came from a fungal species. So you can see many different types of fungi down here in this bottom table. Feel free to pause and take a look at some of these different fungi here. We've got molds, we've got mushrooms, we've got yeast, all sorts of different organisms. And they have a little different common environments. We'll talk about each briefly. And they have slightly different roles, but in general, fungi are decomposers and the organisms that degrade things in the environment. When we look at fungal structures, remember that structure is always important into how we potentially remove something or make something more beneficial to us. They have multi-layered structures, and they actually have a component called chitin. Chitin is actually found in the shells of a lot of shellfish, like lobsters. So it's a slightly different type of polymer. It's a long chain polymer of this compound called N-acetylglucosamine. And that's what these orange little structures look like on the outside. And then those are going to protect the cell from damage, right? The actual structure itself. And there's actually a lot more scaffolding related to fungal structures because they have much more complex structures. So you'll see there's a lot of different glucans and a different type of plasma membrane associated with fungi. And that's because they tend to be three-dimensional, right? They're not these like small little cells like bacteria. So those three-dimensional structures need those strong building blocks. So that's why we see chitin in those polymers as part of the fungal structure. Fungi are pretty diverse. So we can have molds, we can have mushrooms, we can have yeasts. And those are going to be our three main categories for this class. And again, they have those slightly different roles. And this is called a cladogram, if you don't remember this from biology, which shows different what are called clades, which are basically groups of organisms that are very similar to each other. And these just show some of the different fungal structures that you can see, or fungal species, excuse me. So let's talk really quick about some fungal diversity and some different types of fungi that you might come across. The first is going to be yeast. Yeast tend to be unicellular organisms, and normally they ferment under anaerobic conditions. So remember, anaerobic means without oxygen. So fermentation tanks, anaerobic reactors can use yeast sometimes instead of bacteria because yeast do a lot of the same kind of things that bacteria do, but they have a slightly different structure. Yeast are not sporulating. They don't produce spores. They divide through budding. You can have some positive impacts of yeast. So this particular species of yeast is used to make ethanol, which is great, right? We have a big ethanol industry in Nebraska. But we can also have negative impacts from yeast that can cause infections. So as always with microbes, there's positives and negatives, and we want to potentially emphasize the positives and minimize the negatives. When it comes to fungi, we also have molds. So molds are filamentous fungi. So you can see this structure makes little filaments in this particular slide. And each cell in a filamentous fungi in a mold is called a hypha. And the overall structure is called mycelium. So each cell is a hypha, and then many hypha make a mycelium. Parts of the hyphae can grow to produce structures to produce spores, so this is a sporulating type of fungi. And many spores, just like endospores when we talked about bacteria, are very resistant to the environment. Like they're a great place to have important information, right, to protect the organism going forward. We talk about positive and negative impacts, right? Positive here is that penicillin was discovered from a mold. Negative is that a lot of molds can cause lung infection. So if you think about hospital sanitation, we do not want molds in our vents, right? Because they can cause lung infections, especially in patients with pneumonia or that are already immunocompromised. We can also have mushrooms, which of course are what you think of when you think of fungal diversity. They are filamentous fungi that form large fruiting bodies. And so when we look at a mushroom, when you see the cap on a mushroom, that is the fruiting body. So that is the mushroom itself. So there's a large structure that's underneath the ground as well when it comes to a fungi. I don't know if you've ever pulled a mushroom out of the ground, but their root systems are very, very extensive. The fruiting body itself is what's going to produce spores for reproduction. So that has a similarity to flowers in plants. Only the fruiting body is outside the ground. Most of the body of the fungus is below ground. So their root structures are very, very deep. We've actually seen some positive impacts that mushrooms are actually great for holding soil in place because they have those elaborate root structures. So it helps to reduce soil erosion, which is really great for us. But we do have poisonous mushrooms, right? I'm sure you've heard of some examples of mushrooms that shouldn't be consumed. So that's important as well. So a question that I'm going to pose to you is, are we related to fungi? Are we more closely related, are fungi more closely related to animals or plants, and how do we know? So take a pause in the video, maybe write your answer down if you're following along in the slides, and then unpause that video. Did you take that second? I hope you did. So we are actually, as animals, more closely related to fungi than fungi are to plants. And we know because we now use 16S rRNA sequencing to look at what is similar in RNA as opposed to physiological structures. So remember, if we were looking at physiological structures, plants and fungi look very, very similar, right? But if we actually look at RNA, which is now how we do classify organisms, we are actually more closely related to fungi than fungi are to plants, which is kind of interesting, right? I never thought that before learning a little bit more microbiology with you. So we'll talk about some kind of environmental aspects of fungi. So fungi are what we call chemoheterotrophic. What does that mean? It's a great place to pause and to write down what you think that is. So chemoheterotrophic means that the organism uses simple sugars for carbon and energy. So it uses simple sugars, carbohydrates, that are in the environment, and it uses those to produce energy. Now, many times simple sugars are limiting in the environment due to competition. They're a great energy source, so everybody wants to use them, right? So many times fungi have this ability to excrete or secrete extracellular enzymes, which are called exoenzymes, to try and break down complex polymers, because simple sugars are going to be simple polymers. So maybe if they're not there, the fungi is going to need to break down these complex polymers. And so that leads to something that's kind of interesting. It's called a saprophytic lifestyle in a fungi. So let's talk about what that is. Basically, A saprophytic lifestyle is that many fungi are responsible for degrading dead plants and animals, particularly the complex carbohydrates. So the simple sugars are what we want, are what we prefer, but because everybody else prefers those as well, fungi have evolved to be able to go after the more complex carbohydrates. And the complex carbohydrates are in dead matter. They're in animals, they're in plants. And that's basically what has led to a lot of these, what we call saprophytic lifestyles in these decomposers for fungi. So basically what happens is those exoenzymes are released and they are used to degrade that biomaterial, the dead material, and then the nutrients can be absorbed once those biomaterials are degraded. And this is extremely important for waste degradation and recycling. So fungi are natural recyclers in the environment, and we've started to see that it's actually really interesting as a bioengineering application because we can actually use fungi to degrade polyvinyl chloride, which is PVC, which is very, very interesting because I don't know how much you know about plastic, but we have an issue with microplastics and nanoplastics right now that we're very concerned about as environmental engineers. So it's kind of interesting to think about using fungi or using what we know about biology to potentially degrade components. All right, last couple things that I want to talk about when we talk about kind of the environmental aspects of fungi. Mycorrhizae are symbiotic relationships that fungi have with plants. And basically what will happen is the fungi and the plant root systems will interact with each other. And it's kind of a symbiotic relationship where they both get benefits from it. And basically what will happen is it increases the absorptive area of plant roots. So it allows them to take in more nutrients. And plants themselves are a source of organic carbon. So the fungi allow the plants to take up more nutrients that they need for their life processes, and then the plants provide carbon sources in the form of organic carbon for fungi. And so these symbiotic relationships in nature are really, really critical to both species' health. And so if we understand those relationships a little bit better, we actually may be able to mimic them or to use them for our own benefit. Right, so mycorrhizae themselves have this huge surface area for adsorption, and that matters for chemical treatment uptake. So many of the types of treatment that have been a little bit more recent in the field use absorptive principles. I mean, sorry, not absorptive, adsorptive principles. And having a high surface area is really, really critical. So actually studying the mycorrhizae is a good way to understand how do we increase that surface area, how do we figure out how to get that uptake increased. The other thing that's really important here is that nutrients are going to be cycled between the plant and the fungi, so carbon and nitrogen and phosphorus. And there's the potential to increase or decrease that nitrogen cycling or that carbon cycling. And if we understand how that works, we can actually get more productive plants and more productive fungi, which may be a good thing, especially for thinking about agriculture, because we do live in Nebraska. So that's a very quick primer on what's going on with fungi. Make sure that you understand kind of the applications to environmental engineering, and we'll talk about more applications of fungi as we go through the semester.